restart.
Yes.
So, do you want to close this and restart this, or
No, you can start.
just stop streaming and start again, or
No, start now.
and you'll cut it off?
Yeah.
Okay. So I'll just start from the very beginning.
Yeah.
Hello everybody, welcome to the second live session for this course. As was done previously, we have sent you a form to fill in your questions, and some of you responded to that, so we'll start with those questions first. And as you have more queries, please use the chat to send us your questions, and we'll go through that. I'm Rachit, I'm the instructor for this course. Along with me, I have two TAs right here. One is Kamini, and another is Alakesh. Both of them are helping out. Along with that, we have two other TAs that you saw in the first live session. They're also here. So we'll start with this. So maybe Kamini, you can read out the first question.
One person has asked about immuno-isolated cell therapy, wherein they wanted to know how we are encapsulating cells, and we have web vessels also coming into it, and they wanted some clarity into it.
Sure. So as was discussed in the immunosuppressive cell therapy module, so in these are cells you're trying to deliver to be able to perform a certain function which maybe the body is lacking. Maybe it could be diabetes where you have your pancreatic cells not secreting enough insulin, or they're not there anymore. So that's where you may want to use cell delivery. But again, the problem is that if these are foreign cells and the body does recognize them as foreign, they will kill them off. So what is generally done is the cells are encapsulated in such a way that they don't let the immune cells attack them. But then at the same time, these cells are also living objects, so they need nutrients, they need oxygen. So it's actually essential that they have a close supply of oxygen as well as nutrients wherever they are residing. So it's a tricky balance that you want these blood vessels to grow near your implanted device or these cells, but then at the same time these cells at a very close proximity will be protected in such a way that none of these antibodies, cytokines, or cells be able to, or your immune cells be able to come in contact with these particularly implanted cells, so that they can survive that immune assault but still be able to carry on their function and still produce whatever they were implanted for.
So the next question was, how useful is this course in clinical practice?
Okay. Yeah, so I mean, that's an interesting question. In general, I think a lot of these principles that we've discussed through this course are applicable in clinical practice. There are lots of drugs that we've talked about that are actually already being used in clinics, and there are several of them that are currently undergoing clinical trials. This course will help you be able to understand why a certain prescription is given, why a certain route of that prescription is being chosen, let's say oral versus IV versus intramuscular or subcutaneous injections. Or another thing they will tell you as to how this dosing is decided, why doctors say that some drugs need to be twice a day, some drugs need to be once a week. All of that will depend on the drug formulation and how it interacts with the body. Having said that, obviously, a lot of the course that we're discussing is still in the research domain. Not all of that is being used clinically. So, of course, the safety of that, the applicability of that, the efficacy of that is still a question mark. And so, please, whatever you take from this course, just understand the difference between the ones that are in research phase versus the ones that are in clinical phase. And whatever we have discussed in the clinical phase, that of course is directly applicable, and other than that, the concepts from the research phase may also be applicable in clinical practice.
So the next question was to comment on the chronopharmacokinetics of a biodegradable drug delivery system.
Okay, so chronopharmacokinetics we haven't discussed in this course, and neither we are planning to discuss in the rest of the time that is remaining. However, it is relatively a new field where depending on what time of the day the drug is administered, it may have different pharmacokinetics and pharmacodynamics. And that is because our body is in a different state when it's a day versus a night, when we're sleeping versus when we are awake. And so this field that deals with these differences and the pharmacokinetics under these circumstances is termed pharmacokinetics. That we will not discuss too much in this course. In fact, we will not discuss anything in this course. But yes, that is a field. The effects in general do hold true for whatever we discuss. The magnitude may change a little bit depending on the time of the day. Um, but generally speaking, most of the concepts that we discussed will apply regardless of the time that is being administered in.
The next question was to comment on mucoadhesive particles that can be used instead of the particles that were used in a paper published in Nature.
Okay, so I think you guys are referring to the phage therapy paper that I had discussed in the class. That was to use microparticles, which are dry powder formulations and are inhalable, to deposit deep in the lungs. And you are asking whether those particles could also be mucoadhesive. So yes, they could. In this case, the bacteria that we were looking at was external, and when I say external, it means that it wasn't really getting internalized into the mammalian systems or into the mammalian cells. So in this case, mucoadhesive will work also because your particles can then adhere to mucus and slowly release the phage or any drug for that matter. However, just a bit of caution there because if your bacteria is, let's say, deeper inside or some of that has escaped that mucus layer, then you may have transport problems for your phages there. But having said that, I personally don't feel that there would have been much difference had there been a mucoadhesive versus a non-mucoadhesive particle that was used in this particular study.
Okay. So another question which we addressed last time also, which was how is intranasal delivery route used for drug delivery to the brain?
Okay, so yes, we have talked about this in the last live session as well. So generally speaking, delivery to the brain is quite difficult, especially for large molecules. And the major reason for that is traditionally most of the delivery has relied on our circulatory system, mainly blood, to be able to deliver to various parts of the body. However, due to the way the vessels are lined up in the brain, the junctions between the cells is very strong, and these cells I'm talking about the blood vessel cells, endothelial cells, and generally the diffusion of the drug through the blood vessels into the brain is quite low, and that's why most of the drug that you deliver does not end up in the brain, and that's why people have looked for alternative routes. So one of those is intranasal, and it mainly relies on the fact that when you take it through intranasally, the cells that are lining through that intranasal route towards the brain of our body has a very thin layer. It's just in fact at some places just a single layer of cells, and so that just increases the chances of the drug to be able to diffuse. But it still also suffers from the same problems, which is it still needs a small molecule and low doses that will go in. The only advantage there being that rather than exposing all of your body, you end up only giving it locally, and so you can get a higher concentration of the drug as well as you get away with the toxicity that might be associated with it.
Please ask about the questions.
So there was one question asked where they were asking, so if any like drug is given to the eye, so will that interact with the immune system or not?
Okay. Yes. So I think I mentioned briefly in the course, we have some organs that are a bit immunoprivileged. One of that is eye, but so what that means is that the immune response for the same material that you implant in the eye versus, let's say, if you implant it under your skin, those will be different. Under the skin, it will have a much higher immune response than what it will get in the eye. So in principle, you should have lower response, but the response is not minimal, is not completely away. So if you are implanting something that is going to be highly immunogenic, our eye also has blood vessels, and those blood vessels can also cause a lot of immune cells to come in, and that's very apparent if you rub your eyes too hard. You find your eyes become red, and that's because of the injury that you do while rubbing causes a little bit of blood vessel dilation, and all of those blood vessels will be able to carry your immune cells that will come to that location. So yes, you have a slightly higher tolerance for the materials that you can use in the eye, of course, given that they are compatible with the eye cells. But then in terms of the immune response, yes, you have a bit more tolerance, but still it needs to be as compatible as possible to prevent any reactions.
Okay. So the next question was, so let's suppose we give a drug, let's suppose any antibiotic. So they are mainly given to attack the pathogen and kill it. So the question was asked, like, how does this antibiotic or any drug interact with the immune system, and is the immune system then compromised to like self defense?
I see. So yes, sometimes there are antibiotics which may have some implications on your immune system. They may cause the immune system to get suppressed, or sometimes they may even cause the immune system to become more pronounced. But generally speaking, most antibiotics that are currently used, at least so far, not much has been shown in terms of their effects on the immune system. Obviously, there are some changes that always happens, and they could be direct or indirect. Along with that, there are not only just the immune system; these antibiotics can have several other side effects as well. Some of them could cause nephrotoxicity. Some of them causes blindness or deafness. So of course, it needs a very regulated amount of the antibiotics that can be given, and that's where the controlled drug delivery helps, because if you can give it locally, some of these side effects are away. If you can give it in a sustained release manner, then maybe you can decrease the peak amount that you're giving at any point of time, and rather than have a more sustained and controlled amount. So those factors are always in play, which should be considered.
Okay, thank you, Rajit, for the answer. So this is a question from the assignment questions. So some students have a doubt regarding the cascade of events that happen during inflammation. Like, what is exactly the first step which happens during any pathogen attack or tissue damage?
So that's a good question, and we've gone through this quite a bit in the course as well. So let's say I talk about an implantation of a small device, or could be something like a pen. So to do that, let's say I do a surgery first, because it's not injectable. So if I do a surgery, I'm cutting my skin, I'm cutting my underlying fat tissue and muscles. Sometimes if I'm planning to implant it a bit deeper. So all of this causes the blood vessels to rupture. Um, so because as I had already mentioned in the course, we have a very heavy density of the blood vessels everywhere in our body, and pretty much every 100 to 200 micron you'll find a vessel. So those will get definitely severed or ruptured and cause the blood to ooze out. So the first thing that your implant will interact with is all this blood and the surrounding body tissue that may contain several proteins. So some of those proteins may tend to adsorb onto your implanted device, or may at least interact with your implanted device. Once they start the interaction, most tissues in our body have some resident population of immune cells, macrophages, monocytes. Monocytes are mostly circulatory, and generally macrophages are resident in the tissue. So they will start to then survey that thing that you've implanted. They'll start to see whether it's a good thing or a bad thing. If it's something foreign, the body should mount a response. So they will be the first on the site, as well as these blood cells also contain some immune cells like monocytes and neutrophils. They will also come in at the time of the surgery. They will then survey. If they find that the implant is not a threat, at that point they will not do much, and you will be okay with that implant. But if they do find that implant is a threat, they will start secreting lots of cytokines, lots of chemokines, which will then make a concentration gradient in the surrounding location. So these chemokines and cytokines will start to go out, will start to interact with the nearest blood vessels. Lot of the endothelial cells that form the lining of these inner lining of these blood vessels recognize these cytokines, and they get inflamed. And what that means is they start to secrete some molecules, some receptors, which can then bind to the circulating immune cells, and they will cause a dilation of the blood vessel as well. So because now the blood vessel is dilated, and the speed, the velocity at which the blood was flowing in that region has decreased, thereby allowing more interaction between these cells and the circulating cells, and they will start then interacting with those receptors that they have now expressed due to activation with their corresponding ligands on these immune cells. These immune cells will then, through this, they will get a signal that okay, this is the site that there has some problem. Let's go and check it out. So they will start to then transmigrate through these vessels and to the site where the implant was placed, because of course remember that there is a concentration gradient of these cytokines and chemokines, with the highest being nearest to the implant. So they will use this gradient to come and survey that implant, and again depending on your compatibility of that implant, more and more will come, and that's where the cascade of events start. They will try to first see if they can get rid of this device, either degrade this device, or if it's small enough, then maybe just uptake it and clear it out from the system. If they are unable to do so, then the immune response will continue to mount, and eventually, to protect the rest of the body, they will form a layer around it, a thick fibrous layer which contains cells and lots of ECM molecules, to completely wall it off from the system. And at that point, that's where we call it a fibrous encapsulation of your device. And if let's say your device was supposed to measure glucose, because of this capsule now it can't really interact with the glucose present in the surrounding, so your reading you'll get is erroneous. If the device was containing some cells which were supposed to secrete something to complement whatever factor you were lacking, that cannot happen, because whatever the secrete can't go through. Not to mention the implant, since it's already surrounded by a thick capsule, it doesn't have access to nutrients and oxygen, and so those cells will also die. And so that's basically, in a nutshell, the cascade of events that happen. And more details you'll find in the slides. There are all kinds of molecules and cytokines and receptors that I mentioned there. So you can go into that slide if you want further knowledge on this.
Okay, thank you, Rajat. Next question is a slight more in-depth knowledge on the EPR effect. So a lot of students had a doubt on the EPR effect, like how does this function, and
Okay. Okay. So maybe I'll use the thing here. Okay. So question is on the EPR effect. And as if I use the full form here, then EPR is nothing but enhanced permeation and retention. Now let's look at each of these terms individually. So let's look at first enhanced permeation. And so what this effect is saying is, in cases where there is some inflammation at the site. So I just described how with inflammation you have more dilation of the blood vessels. So let's say I have this blood vessel that is going through a tissue, and for some reason this tissue has inflammation, either because there's some bacteria or some pathogen, or maybe there's some tumor that has been growing in this area and the immune system is trying to get rid of it. So some inflammation exists here. Now because of this inflammation, and as I was just describing, this blood vessel is becoming leakier. And what I mean is, earlier there were cells here, now that it's dilated, it's become bigger. So these cells are slightly further apart. So instead of being like this, they are closer to something like this. And because of that, the see if it rubs. Okay, it does. Okay. And because of that, whatever is flowing here earlier, let's say if in these vessels anything below 5 nanometer could pass through, but not above that. So anything greater, less than 5 nanometer was able to go through, but anything above 5 nanometer was localizing in the blood vessel itself if you injected something IV. Now, because of this, maybe you can even get 50 nanometer to pass through. So that means that if I inject something between 5 and 50 nanometer, I will have a much higher localization of those objects or those particles in these regions than compared to my healthy tissue. So let's say this was healthy tissue. So this is enhanced permeation. Now, this was in terms of inflammation. In tumors, of course, you have a large cell mass that is now trying to grow, and because it's trying to grow, it's recruiting new blood vessels. So in tumor, not only because of this inflammation, you have dilation of the vessels, you have these new vessels that are also formed. And these new vessels are extremely leaky because they're quite immature. So they were formed very rapidly because the tumor grows very rapidly, and so then they didn't have time to mature. They didn't have time to get a very uniform lining, so that only things below 5 nanometer can pass through. And because of that, they are also leaky in general. So their gaps could be even higher. They could even allow, let's say, even a 200 nanometer object to pass through. And because of all this, anything in this size range between this 10 nanometer to 200 nanometer that you inject will tend to localize in inflamed tissues and tumor tissues. So that's enhanced permeation. Now, what do we mean by enhanced retention? It's the same logic here. Now, most of our tissues have a parallel circulatory system which is called lymphatics. And what is the major job of lymphatics is to basically collect any extra fluid that has been flowing through your system and then return it back into the major circulatory system. So it kinds of act as another drain that takes care of any extra fluid. Now, since my lymphatics hasn't changed a whole lot because it got inflamed or there was tumor and grew rapidly and recruited new blood vessels, but they didn't really focus that much on the lymphatic system. So the capacity to clear the fluid is limited. And because of that, there's a higher fluid pressure in this tissue than in any other tissue. So at that point, there's some fluid pressure that this tissue holds. And so whatever objects are coming in into this inflamed or tumor site are basically not able to diffuse away, move away through normal processes as they would in other tissues. So because of that, they start to accumulate here. So first of all, they're accumulating because the blood vessels are leaky, and so your input is increasing, and then whatever was the output of these, that has slowed down because the lymphatics are not good enough, and all of that compiles to form this effect, which is called enhanced permeation and retention. And this is used quite a bit in tumor application where people will deliver things of 100 nanometer, 200 nanometer, and expect that to accumulate more in the tumor region. In fact, there's already clinical products for this. We have discussed in the class. Epr is one of them, and Doxil is another one of them that uses this effect along with other sustained release that happens also helps. But that's the major process that goes through. Okay.
Okay, thank you, Rachit, for the answer. Next question is, so students want a bit more uh, sol- explanation on the Broman effect.
Okay.
So,
Okay. So the Broman effect, as we discussed in the course, were mostly for protein adsorption. And so let's say if I have a surface, it comes in contact with fluids containing proteins and other biomolecules. So generally speaking, these proteins need some time to diffuse and come to the surface. So and let's say this protein contains a mixture of proteins. Let's say thousands of different types of proteins are present in this fluid, which is generally the case when we talk about any body fluid. And so because now there is a time lag between all the proteins to be able to come and explore the surface, uh, whatever is the protein which is nearest to the surface will be able to interact with the surface first. So those proteins then interact with the surface. As we had discussed, these could be hydrophobic or hydrophilic surfaces. Proteins are weak surfactants, so they can change their structure and try to reorganize them to be able to interact more with that surface, form van der Waals forces, ionic bonds, hydrogen bonding. All of those are feasible. So they will then start to form all these bonds and first attach and adsorb onto that surface. Now, maybe there's, so let's say this is protein A that has done this. Maybe there's another protein B that can do that, but with a higher affinity. So, but then that protein B was not immediately in the vicinity. Maybe it took some time. It took maybe a few seconds for that protein to come and interact with the surface. But now, because it has slightly higher affinity than protein A, when whatever bonds it form are a lot more stable, and because of that, it will eventually, over a period of time, and that time could be over a period of few seconds, could be over a period of few minutes, or could be longer, that protein will start to replace whatever was the first incoming protein, that was protein A. And because of this, what you will have is eventually the protein that were in higher concentrations will attach first, and then they will be replaced over time with proteins with higher affinity. And this is the Roman effect. And this whole effect of higher affinity proteins being able to attach and replace the lower affinity proteins is Roman effect.
Any more questions?
There was one question which was asked wherein they asked about the challenges related to the lack of knowledge about the human physiology in the field of drug delivery.
Okay, so this is an interesting question and actually quite valid question. So generally, a lot of research has been done on understanding human physiology. Still, a lot of research is going on. So there's quite a lot that we know about the human physiology, but there's also quite a lot of literature that we don't know about the human physiology. So, of course, if we want to model this, if we want to figure out how much of the dosing we need to give, we need to know. Ideally, for a perfect model, we need to know everything, and only then we can design the best system suited for that particular body or for that particular disease. So yes, it's a valid point. They have physiology. The more understanding we have, the better we'll be able to design these systems. To give you a very recent example, about 2-3 years ago, they just discovered for the first time that the brain has lymphatics. So now, if that system was not known previously, then we were not even talking about clearance through lymphatic systems. But now that we know that there's lymphatics, and that could be modeled in, that completely changes of the dosing and the amount that could be given, as well as how these things clear out from the body. So obviously, more in-depth knowledge of physiology, more discovery on physiology will go hand in hand and help the drug delivery field to design better models, to design better systems that will eventually lead to better patient outcome. So yes, physiology is definitely important and quite a lot that it can contribute to this field.
Okay, there was one more question which was asked: how can gene therapy can be used in vaccination and it can be brought into the clinics?
So gene therapy and vaccines, I think we'll discuss the whole module if we haven't already. I believe we have, but I'll just discuss it back in a nutshell. So so let's say rather than, and let me go back and tell you where's the need for this. So as we were earlier describing of why people would use cell encapsulation and immunoisolation is because if something is been lacking in a certain system, we want to complement it. So the example I gave, let's take that example again. Let's say it's type 1 diabetes. My immune system has reacted against my cells, and that has caused all my pancreatic cells to die, which means that I can no longer produce insulin. And that is quite severe because every time the patient eats, its blood glucose level is going to go off the chart, and that may cause problems like infections and other associated issues. So at that point, I want to implant cells that may be able to complement this insulin production and be able to give a healthy life to the patient. But now the problem is that the immune system is constantly pounding on the cells. Maybe these cells are derived from another human, and since these are derived from another human, those human antigens may act as a stimulus for the immune cell to kill these implanted cells. These could be derived from some other species altogether. Maybe these are pig cells, maybe these are cells derived from some other organism, monkeys. So those will generate even higher immune response. So that's where people started to say, what if we just modify the cells that are part of our body that immune systems know that these are cells, won't attack them at that rate as it does for other cells, leading to a longer survival of these cells. Of course, they may still attack it because let's say if I put an insulin gene, then since these cells are producing insulin, insulin at the first place was anyways reactive, so these cells may still attack those cells, but the immunogenicity is much lower because it's at this point only the insulin that is being affected, and in some cases maybe that is not present any longer. So at that point, the idea came that let's see if we can just give the gene itself and use this. And when we started doing this, then of course we saw that gene therapy itself has its own challenges, but then it may cause a lesser immune response compared to cell encapsulation. But then now coming back to the second part of the question of using them as vaccines, that is again a further byproduct of this. Rather than putting the whole organism or whole pathogen into your body, which sometimes some of these humans may not be able to cope up with. We decided why don't we produce these antigens from the body itself, because the whole point of the vaccine is let the body get trained against an antigen so that when the actual antigen comes, the body is absolutely ready and doesn't let the disease to get established. It clears it off even before the pathogen is able to colonize the body. So that's where gene therapy came in. One thing that came in further here was our immune system can be majorly categorized into two types of responses, one against extracellular proteins and one against intracellular proteins. And the advantage was using gene therapy was that everything you make basically acts as intracellular because this is being produced by the body itself. It's in the cytoplasm, and so because any external antigen you give, more often than not, that antigen ends up being in the endosomes, which triggers a different type of immune response. So all of these combine together to cause this field to come up, and that's where the gene therapy for vaccines has come up. And then, again, I'll discuss this in much more detail as we go along.
So in one of the assignment questions, we had given a choice that the basophils will be not an antigen presenting cell, but recent papers have cropped up showing that they can be used as an antigen presenting cell in TH2 mediated response. So should we address that question?
Yes, I mean, so more and more, as I said, these are general concepts that we give in this field. More and more research is being done. We just answered some of these questions about physiology, that more is discovered. Similarly, a lot of research has gone into different immune processes. In fact, immune system itself is a very, very dynamic field. That every 5 to 10 years, it completely changes itself. So yes, I mean these things do come up. What I'm trying to give you through this course is general concepts of which are the major antigen-presenting cells, and those still remain these macrophages and dendritic cells. Of course, some papers have shown, on the contrary, that some other cells can also act, which is something that the field will evolve and the course will update as we go on. So just take that in that spirit, please.
Anything else?
So please feel free to write your questions. If you have, we are almost out of the questions that were asked in the form as well as that were discussed in the discussion forum. So if you have, please write it now, or we can stop at this point. You as PAs have any questions while we give them another 2-3 minutes that are often asked.
So we can inform them about the certification. They will
Yes, there was some problems with the course registration for the exam, but that has been sorted out now. So the link that is now present or has been sent out, that completely works. So if you are interested in registration for the exam to get certification for this course, please feel free to do so. I'll just discuss the exam format as well, because there was one of the questions that was asked in the last live session as well. So these will be predominantly multiple choice questions with more than one correct answers. In these cases, if there are more than one correct answers for a question, and if you only select one, you only get half a point for that answer. If you select a wrong answer along with the correct answer, you get no points. Whereas if you select every correct answer, then you will get the full point. So just make sure that whatever you select for only the correct ones. Whether they are one or two, it doesn't matter. And then along with that, we'll have some numerical type questions in which you may have to enter just the words or just the numerics. So the answer could be 24 hours. So we'll mention in the question at what units you need to mention it. So you just need to mention 24 there, and that will be acceptable within a certain range. So maybe the range for that answer might be 22 to 26. So all within that range will be considered correct. Yeah, so that was that. Another thing that in the live session last time somebody had asked that whether we can show some demonstrations in these live sessions. Unfortunately, most of the things that we deal with while doing the synthesis is quite big, and it's not really possible in this setting to be able to show those demonstrations. I do believe there are some courses on the NPTEL that are lab courses, and then where you do see what goes on in the lab. So I encourage you to see that. There are some YouTube videos on these processes as well. You can see that, but unfortunately we won't be able to show you a live demonstration because not only these are large objects that are difficult to carry, but some of them also has safety requirements that may not be able to do in this setting. Okay, looks like there are no further questions. So in this case, we will stop right here, and we will have another live session. We'll announce the date as always. So look forward to more interaction during that live session. Thank you.